Hi guys, Dr. Orlando here with a video on non-disjunction during meiosis. We're going to look at what happens when chromosomes fail to separate properly during meiosis 1 and meiosis 2. This is a picture from your book, so you can look at that. It's figure 912, and I'm just going to go ahead and go through it and describe it as we did in class instead of trying to draw this thing out. Okay, so a non-disjunction, like I said, is the failure of the chromosomes to separate properly. If they don't separate properly, then you end up with daughter cells that don't have the right number of chromosomes. So let's see what would happen if we make daughter cells with the wrong number of chromosomes and those daughter cells are able to be fertilized. What does the resultant zygote look like? All right, so I'm just going to go ahead and label a few things here and then we'll get going. All right, so this whole process is showing meiosis and fertilization, and the two phases where the chromosomes separate is anaphase 1 and anaphase 2. So this first slide that we're going to look at, and then we're going to go to the next page, is going to look at what happens when there's a failure of chromosomes to separate during meiosis 1. And in fact, this is the one that is most common in humans and mostly responsible for changes in chromosome number. And it's because, we'll see, because it is a failure of chromosomes to unpair. So we know that during prophase that homologous chromosomes will pair up and exchange some of their DNA during crossover. And if those chromosomes, homologous chromosomes, don't unpair, then they may travel together instead of splitting during anaphase 1. So let's take a look at that. So here we are at anaphase 1, and what should happen is one of these homologous chromosomes should go this way and one of them should go this way, right? Now that doesn't happen. Okay, this should be labeled meiosis 1. Okay, non-disjunction at meiosis 1. So that pair, instead of splitting, goes to one particular cell. Now they're not showing the entire, all of the chromosomes. They're only showing the pair that doesn't split. Okay, so this pair didn't split, so this cell ended up getting both of the homologous chromosomes, a pair of homologous chromosomes, and this cell didn't get any. Okay, or at least didn't get any of that pair. It got all the other ones that it was supposed to get. It didn't get that pair. So if we follow this guy that didn't get pairs, didn't get his homologous chromosome that he was supposed to get, okay, meaning he's missing one, right? So as we go through meiosis 2, there's no sister chromatids to separate because the homologous chromosome never showed up, so we're still minus there. And then finally, when a sperm comes along to fertilize that egg, what happens? This sperm is bringing the chromosome, and it has no homologue to pair with. So this zygote then is minus a chromosome. So we say that this is, if we were to say what's its karyotype or what's its genetic, its chromosome number, we'd say that it's 2n minus 1. It's missing one of its homologues. Okay, let's look at the top picture here. So this one, he ended up getting, should we say she, since these are the eggs, she ended up getting both of the pairs, both of the homologous chromosomes. And when they went through anaphase 2, they split properly, as they would, so that the sister chromatids are separated, right? So now they've got an extra one. She should only have one copy of this pink, right? That guy's supposed to It's supposed to be here, but it's not. Okay, so we've got two copies, and I shouldn't say copies, two versions of that chromosome. Let's say this is chromosome number 21. Okay, so we've got two chromosome number 21 in this egg. So then here comes the sperm carrying another chromosome number 21, and your next thing you know, we've got three copies of chromosome number 21. So we would say that this chromosome number is 2n plus 1. It's got all of the chromosomes that he's supposed to have plus 1. Okay, once that sperm comes along. Okay, if it is chromosome number 21, and I'm not just pulling that out of the air, this is the most common trisomy. It's the most common trisomy, meaning there are three copies of one of the sets of chromosomes, and this is Down syndrome. Okay, and again, the most common of the trisomies in the autosomes. Remember the term autosome? Autosome refers to chromosome. In humans, it's the chromosome 1 through 22. So it's all of the chromosomes that bring us information about regular body cells, whereas the sex chromosomes in humans, this is pair number 23, and it gives us information about about male and female, along with other things. But remember, females are XX and males are XY. So that pair is occasionally, 50% of the time, it is an unmatched set, being an XY. Okay. Either way, if there is a non-disjunction, we can end up with plus or minus a chromosome here. Okay. All right, so that's failure, or that is non-disjunction during meiosis 1. And you can see that all of the daughters would be affected. So that's a key characteristic in meiosis 1. Non-disjunction in anaphase 1 causes all of the daughter cells to have an abnormal number. Okay, so what if there's a non-disjunction during meiosis 2? Now this is less common in humans, but still certainly possible. Here's what we see. So here is the egg going through meiosis 1. Homologous chromosomes will line up on the equator of the cell, the metaphase plate. They will be separated from each other. Perfect. Did it exactly the way they were supposed to. So now we go into meiosis 2 and anaphase 2, where the sister chromatids line up on the equator, and the sisters get separated from each other, just like so. Okay, perfect. Good job. Each sister went to a different nucleus, and should a sperm come along and fertilize one of those eggs, then we have a baby here who is normal. They've got two copies of each of the chromosomes that they are supposed to have. So these guys are going to be perfectly fine. Okay. So then let's go down here to where they have our little yellow dot indicating that something went wrong. So, that something went wrong during this meiosis, and sisters failed to separate properly. They failed to go to the proper poles. So one of the chromosomes that was supposed to move to the poles went to the opposite one. So you got two of them on one end and 0 of them at the other end. So when the nucleuses reform around them, you would end up with an extra chromosome here. Okay, extra chromosome there. When the sperm come along and fertilize it, now this one would be a 2n plus 1, right? And this one would be a 2n minus 1. Okay, so both of these would be abnormal and could result in would result in chromosomal abnormalities if they were fertilized. Okay, so non-disjunction during meiosis II would only affect the cells whose sister chromatids didn't separate. So this one that went along was just fine, so 2 of them are fine, and then the one where the sister chromatids did not separate, 2 of them are not fine. Alright, so that's our non-disjunction. If you have any questions, please feel free to contact me.